Oh, thank you very much, and thank you for inviting me. Since this is near the end of the meeting, it's occurred to me, listening to all the talks, that there's a lot of information out there. And I'm trained as a geneticist, and one thing you have to be able to do as a geneticist is stand a great deal of ambiguity. So when you early start doing genetics, nothing makes sense. And if it bothers you, you should quit. And sometimes you have to hang on to that ambiguity for years, and it'll all sort itself out. So looking at this meeting, you might think, oh my gosh, there's all this stuff. Yeah. And our job now is to figure it all out and make sense of it. But more importantly is to take that information and find ways to do treatment. And we should be able to do treatment before we understand it, hopefully. We don't have to wait for that. Now, I got involved in this study. Well, let me just, I want to show you my motivation. This is my son. He's been missing for 5 years. I have to do this research. I was also on the IOM panel. We met for about a little over a year and a half. We looked at 9,000 publications. That's every publication that was existing at that time that mentioned ME/CFS. Many of them only mentioned it, and that was it. My job was to find the molecular data in that set of 9,000 papers, and I was a little shocked that I found very little. And my task was to find a biomarker amongst all those 9,000 publications. I couldn't find it. That set me on a path saying, well, we have a problem because we don't have an awful lot of molecular data that a field will need. And so I decided what I had to do was really to collect data. Now, the scientific method is to make an observation, and then from those observations, make a hypothesis, design a test to test the hypothesis. The test will then rule out or rule in the hypothesis, and then you make, from that are new observations. And from those new observations, you make another hypothesis, and it goes on and on and on. The problem is it starts with an observation. The problem with NIH that they have is that in the early days of NIH, they had a large number of trial and error research. Let's try this, let's try that, with no idea of what it might even do and would it even work. And they got rid of all that, because it was very inefficient, by saying, no, you have to start with a hypothesis. And that's great, and it really improved the quality of the research. But it left a hole. And that hole is, how do you start a new field? There are no observations. You can't make a decent hypothesis, really. And so you're stuck. So, in fact, I realized that we needed a lot of data, so I wrote a grant, or in fact, wrote 2 grants, but they were both not accepted because there was no hypothesis. So, solving that, I teamed up with Lyndon Tannenbaum at the Open Medicine Foundation and raised private funds to do the study. The idea is that all the data that we generate will be put up on a web that all the scientists can access. We'll try to do that even before publication because we want to get this thing rolling. And we'll try to make it easily accessible and people can download the data. And we're working on the software to make that easily done. Now coming back to, so the 2 purposes of what this project was going to do is one, collect a lot of data. The second one is, in the collection of a lot of data, try to find biomarkers. We don't really know where to look. And, but one thing we can do in this search by collecting a lot of data is to look in lots of places for this biomarker, but do it all at the same from the same time point. What you really want for a biomarker to be very effective is to find a molecule or a group of molecules that are created by some process, and an independent molecule or molecules that are generated by a different process, but both of them caused by the disease. If you have at least two, you're going to wind up with something that's very, very effective. With one, you can have some problems. The patient has some mutation, something a little different. It doesn't work very well. So that's the idea. So you can do that if you take them all at the same time point. And it also means that you don't have to use all the same type. We don't have to use a bunch of metabolites. We don't have to use an RNA molecule or a protein. We could use a protein and an RNA because they're done at the same time. And when you do the diagnostic test, you're going to take the sample at the same time to do both of those. So this just gives you a little bit more power. And the way an awful lot of stuff has done is that people study the proteins, or they study the RNA, or they do the DNA sequence, and you can't combine them. So it's more effective to be able to combine them by taking the same time point. But doing all this is very expensive. And I think we're spending about $70,000 in data collection per patient. And if we wanted to do 300 patients, even that would be expensive for NIH. It would be astronomical, really, in an expense. So how do you solve that problem? One way to solve that problem is look at the severely ill patients. And it's useful as well because they have hardly ever been studied. What we see from a lot of other studies is that severe patients with a disease usually have larger molecular signatures. If you have larger molecular signatures, you don't need as many patients to get statistical significance. And so that reduces the cost. Now, of course, what you do after that is if you see where to look in the different things, and now you go back to a bigger population, possibly not severely ill, and look at a much more limited set of things, and that'll be less expensive. So it has to go in these two different phases. So for this project, I'm not going to go into detail about these. The project has been started. We've identified the patients. The samples have all been collected from all the patients. We now decided to do 20 patients. And we're going to do 10 controls. Now, we're doing less controls than patients, and that's because it's very likely that the variance in the patient population will be greater than the variance in the control population. And there's no reason to add controls just for the sake of adding controls, because it costs us just as much for control as it does for a patient. I was involved in a very large trauma study, and we looked at 5,000 trauma patients. And we had to do controls, and we wanted to statistically figure out how many we needed to do as we started to do that, and it turned out we only needed 20. 5,000 patients and 20 controls, and that's all we needed to get the statistical significance because there was tremendous impact of trauma. So a lot of this is going to be dealt with in terms of sequencing. We're very good at that. We've worked out a lot of the technologies for doing sequencing. A lot of the cost has come down. A lot of my students are now running companies that do sequencing. I try to make them do stuff for free. It's a payback. And they're very generous. I've had no problem with that. They're very generous at doing things for free. We also have to do immunology. I am not an expert in immunology. I would not trust my judgment. And so we got Mark Davis, who is a superb immunologist at Stanford, chair of the immunology department. He's heavily funded by the Gates Foundation. He will take charge of all the immunology. So another style of this is not to come into one laboratory and try to do everything. We want to do things we know how to do well. And so one of my jobs is to find people that are really good at what they do. So there's a lot of expertise in my lab, and that's the Stanford Genome Technology Center. Also, in fact, one of my past students is Mike Snyder, who's now chair of genetics at Stanford. He's also, we think, an awful lot alike, but he's set up to do an awful lot of things that we're not set up to do, so we don't have to buy any equipment. So this whole project is going to be done without buying any equipment. And it'll just be going to people who already have the equipment. We have developed some new devices. I'm not going to talk about, I talked about those yesterday. We're very good at developing devices. We're very good at decreasing costs of things. When we develop a new technology to replace an old technology, we always try to get at least 100-fold cost reduction. One of these little devices costs 5 cents per assay. One of my students also built a cell sorter. Now cell sorters cost $150,000. He built one and took it out of his pocket. It's all made by inkjet printing and three-dimensional printing, and his cell sorter costs 10 cents. And it's actually disposable. So those are the kinds of things that are around the corner with technology, and it's really snowballing because once you realize how you can do cost reduction, you can continue to do that. So all of these technologies we've had to do pilot studies on. Many of them we've done a lot of. We don't need to do pilot studies. But some of them we haven't done that much. And so before we launch into valuable samples, we want to make sure that everything works okay. So one of the things that we wanted to do is metabolomics, and a couple of talks have already talked about that. It's kind of new. There are a bunch of mass spectrometers, we have one, but we're not really professionals at metabolomics. So we had to find another way to do that. And we teamed up with Metabolon. I knew their existence. I tried to get them to do this about 3 years ago. They said they had the instruments, they were ready to go, but the software wasn't ready. So they called me at one point about a year ago and said they're now ready. And in fact, my son was one of the first samples they ran for a disease. We've also now teamed up with Robert Navio, who is an expert in both metabolomics and mitochondria. He is an expert in mass spectrometry. So he's going to be part of this big data study as well. In fact, because I think the metabolites are so important that we're going to do it both. We're going to do it at Metabolon and with Robert Navio. They do it differently. They use different collection tubes. They see different things. Metabolon does about 500. Bob Navio, his work will add about probably another couple hundred unique ones, plus a lot of overlap. So we'll wind up getting about 700 metabolites that we can see. We would like to see them all. Eventually, the technology, I think, will get there because we have to do that. Now, there's a problem here with these little pilot studies. Is that because how many we're going to do for our pilot? We're going to do 3. That's not a big study. But you've got to keep in mind what we're trying to do is personalized medicine or precision medicine. That's 1. So if we can't see it in 1, it's not worthwhile. But you want to make sure that it's reproducible, and that's why you need more people. But you can still look at 1 and see what you can learn from it. And we also needed controls. So this is probably not quite kosher, but Metabolon had no controls, and we absolutely had to have them. They ran my son's sample, and but we don't have what's healthy. So they got all their employees to line up and donate. That's not normally how you do things academically, but in fact, I love that because it instantly solved the problem and we got the data. And I think I'm very in favor of don't do anything unethical, but do it fast. Now, also, it's interesting because when we looked at their patients, in fact, so some people, I think, were talking about this problem that when you do controls, well, what have they been eating and what medicines are they taking? You need to take a record of that. Well, not really. With this technology, I can tell what they're eating. I can tell what medicines they are taking. So as doctors, you don't have to worry about that. Some are sliding something aside. It shows up. If they're taking drugs, you find out. So it's actually going to be very versatile in the medical community. I think it's going to be standard. I think people should do this at least once a year, maybe more often. So most of this particular work is coming from the serum. We haven't yet done the cells. We're going to do that. We're lining that up now to do various cell types. But right now we're just doing serum, which is sort of an averaging of what's in the body. We don't know where the sources of the metabolites are, what cell type, what organ, and so forth. So it's all an average. You've got to keep that in mind. And it's not in cells. It's outside the cell. So, well, before I jump to that, the biggest problem we have in all of this stuff is you have a massive amount of data. How do you look at it? Now, what normally is done, you make a table. And in fact, we did in our trauma study, we had a lot of data. We had about 2 billion data points. So one of the surgeons there said, I don't like all this bioinformatic analysis. I want to see the data. And we said, it's too much. He said, I'm going to believe anything you say unless you show me the data. So we brought in a stack of printouts about this high and set them in front of him and said, here's the data. And he looked at it a moment and he says, I get your point. So that was one of my first lessons to learn when I started doing major DNA sequencing. You always want to see your data. You can no longer do that. It's too much. You have to find ways to help you interpret it. And so when we have all these different metabolites, we've been searching for ways to actually to be able to see it quickly. And so this first slide is going to be something you can't see very well. It's only meant for you to get an idea of the impression of the data and how we interpret stuff. And this is working pretty well. So here's the metabolism of the human being. What's wonderful about this is it's almost all worked out. Not last year, 30 years ago. Most of it's probably right. And it was done with test tubes and pipettes and so forth, the old-fashioned way. But here it is here. Of course, you can't really see it, nor can we. We put it on a computer screen. We can't really see it either. We use a big screen. But what can rapidly do is zero in on it. And then we can do this zero, and we can move to there, and we can move to there, and we have some codes that we can say, okay, let's look at the citric acid cycle. Let's look at this. Let's look at that. And it's very fast. But we needed to color code it to do something to help us see it. So in that color coding is if it had a much higher than healthy controls, we colored it red. If it was much lower, we colored it blue. Now, the way this works is that you have a metabolite and it gets converted to something else. And then there's an enzyme that makes that conversion. And then this one is used for something, and in fact it's used for two things. And so it'll go off this way, and there's an enzyme and a resulting thing, and it will go off this way with an enzyme and then a resulting. And it's just a big network. And sometimes it comes all the way around here again. And you can see that, and that's what these lines are the connections. The circles are the concentration of the metabolite, and the little squares are the enzyme. Now, the software we've set up allows you to click on any little point. It will tell you what that metabolite is, or you click on the little square, it'll tell you the enzyme. And then, in fact, it will tell you a lot of references. And you can click on the reference and you go right to the data. So it's very fast to navigate. So you can see in this already, if you're not, we set this up as red and blue, so if you're colorblind, you can still hopefully interpret it. If most people use red and green, that's a disaster. And you can see lots and lots of blue, and you can see lots of red. And so what struck us right away when doing the metabolomics of CFS patients, there's a lot of differences. And much, much more than we see in almost any of the techniques we've looked at. Now, when we color these things red or blue, we're not talking about 20% more. We're talking about something statistically that's 5 standard deviations away from normal. If you know anything about statistics, that's a lot. And so one of these, in fact, is my son, and it's 16 standard deviations lower than healthy control. So these are big differences. And it's hard to understand how they couldn't have a huge impact. So if we then, I'm just going to go through a few examples. We only have a few patients, and there's a couple of differences in them. You can learn something from these. And it'll give you a little feeling. That's what I'm going to try to do. This is all about getting a feeling. You don't have to understand any of the names. It's irrelevant. We have to make up names, and they're almost ungodly complicated names. But they're unique. So there's nothing else like it. It's unique, and that's what's nice about it. So here's a pathway that we see. And what you can see from this pathway is it is a circle, and it's something called the citric acid cycle. This is where most of your energy comes from. This is inside of the mitochondria, and it's how you burn the carbon, basically, by the oxygen, and you give off carbon dioxide. And this is where most of your energy comes from. If you put on one patient onto this with color coding, a lot of deficiency. This one is citrate up here. I can hardly read it. One of the big ones down here. Oh, the size of the circle is proportional to the surplus or deficiency. So if it's a little circle, it's not very bad. If it's a big circle, it's really bad. So again, we're trying to use visual coding here so we can very quickly look at it and figure out what's going on. So we look at this very quickly. We say, oh my gosh, there's a real problem in the citric acid cycle. This is succinate. That's a really important one, and it's very low. Now, what we also can see, and I'm not going to drag you through all these things, is there's basically sort of 3 ways you can generate energy. You can burn glucose, and you burn glucose in the citric acid cycle that enters up here and goes around and around and around like a wheel. You can also bring in fats. And the fats will go into this cycle and go around and around and around. They're using different chemistry. They come in on the cycle over here. And you can also burn amino acids. And they'll go into the cycle as well and generate energy. So that's a wonderful protection for people. Because from carbohydrates, you can make glucose, and that's in your diet. You can store a little bit of that. But you can also use fat, and you can store fat, and some people store a lot. And it's a total different molecule. It's very energy rich. But if you run out of fat and glucose, you can burn amino acids. Amino acids are how you make proteins. And you have a reserve of amino acids, and it's called muscle. So when you go into starvation, you'll burn your fat first, and you'll burn your glucose first stored, and you'll burn your fat, and then eventually you'll start burning your muscle, and your muscles will shrink. And that's important for this, because when we look at it, it would appear to us that glycolysis does not look like it's working very well. Glycolysis is how you burn glucose. That's how you normally make energy quickly. Cancer cells almost exclusively burn glucose to grow. It doesn't seem like it's working very well. If we track the carbon, the glucose goes away. And it's made up of carbon and hydrogen. And where does it go? It looks like it gets converted into fatty acids. And it looks like it's probably stored because it can't burn fat very well either. And so what we've looked at in one of these technologies is the density of the cells. All the white cells are light. And you know that oil floats on water. And if you're very obese, you'll float very nicely. And so I think they're converting their glucose into fatty acids, and it's simply being stored and not burned right away because the cells are quite light. But it looks like the patients are burning amino acids. And that might be useful in terms of even some thinking, in terms of thinking. Now, proteins are hard to digest. So if you have digestive problems, it may not be convenient to do that. But if you were to eat some very simple proteins that will convert to amino acids, you might get more a feeling of energy out of that because it looks like it's more readily burned than the others. Don't know for that, but it's something that's a very easy test. I know when my son, when he was ill and we were trying all sorts of things, he accidentally discovered that he felt better when he ate amino acids. And he was very stoic. So he would take spoonfuls of a mixture of amino acids and just eat them. And I tried it. It's awful. Absolutely awful. And he would sit there eating his porridge, spoonful after spoonful of amino acids. And I said, doesn't it taste terrible? He says, yeah, it's awful. But he got more energy out of it. So these are just some of the compounds. I'm not going into detail of this. This is actually my son. And one of the things you can see right here is asparagine. That's an amino acid. It's extremely low. And you need it to make protein. And so I was desperately trying to find a way to get asparagine into him. But it's not that there's more that are low than high, but there are definitely some things that are high. And it seems that some of this is high are involved in, and maybe in some of the fat metabolism because it's not being metabolized very well. I'm just going to show you a couple of things here. Don't worry about the names. The names are irrelevant. They just have to be unique. But looking at this, and we know biochemistry, that made it interesting to us. And then showing you this on the pathway up here, these are the ones that are very high, and there's some things that are very low. What's going on here? It shows this is an indicator that he is deficient in biotin. That's unusual. And you talk to doctors about it, nobody's deficient in biotin. The microbes make it. Well, it turns out the microbes in his gut weren't making very much biotin. And it was an easy thing to fix. Give him biotin. Really easy. The problem is you don't know it. So I'm guessing here with a lot of this kind of technique, you'll find, individuals will find that they're deficient in something that's very easy to fix. Looking on, we got an email from somebody because we sent an awful lot of emails. There was one patient that started taking biotin, and it totally turned him around. I'm not sure that's common. But it's a wonderful when you find something that's very easy to fix, and your problem is you don't know. So what I like about this technology, it will allow patients to know a lot. Now, I don't expect people to run out and buy their own mass spectrometers. These things cost about $1 million. And it's not even clear that every laboratory needs to have it. I'm perfectly happy having Metabolon run them. So far, they've been running everything for free. We're going to pay them to do this big data study because they've been so generous. And Bob Navio is also doing it, and he's an expert in this stuff. He knows a lot more than I do. But I'm excited about this technology, and I'm excited about what it's sort of suggesting to us. So just these few patients say it looks to us that there's a real problem in the fundamental energy generation. It's not just a mitochondrial problem. Stuff is in the mitochondria, but the glycolysis is burning glucose, that's outside. Now, what's also interesting is that when I was involved in this big trauma study, is when you get hit by a bus and you don't die instantly, your mitochondria shut down. I don't know why. So it would appear to me that it's a natural process of some kind of protection. It's possible in a crude sort of way that it shuts down to keep you from moving. If you have a big trauma event, you probably have broken bones. If you move, you might puncture a lung. So shut the mitochondria down and you're not going to move. And I think everybody and the patients in this audience know that that will shut you down, right? And you won't be able to move. Now, to me, that's exciting because it may indicate that this is not some foreign process. This is not necessarily an infection. I'm not suggesting, I'm giving you hypotheses that have to be tested. And I'm just adding one. A lot of people have been looking for infectious agents. Absolutely have to do that. But it's also possible that it's totally natural. The problem is not that you got shut down, that's natural. The problem is you don't get restarted. And that's what we have to focus on. Why don't you restart? If you have a major trauma, your mitochondria shut down, and then they start up again. One hypothesis that we have about that is that by seeing all these metabolic deficiencies, you may have a number of them at the time you had the big event that started it, the infection. And you have too many deficiencies to restart, and your body got locked in. The reason I'm saying that could turn out to be fairly easy to fix. We just have to know what to do. And we're developing, community-wise, developing lots and lots of tools with people doing different things that I think we're going to figure this out. And it could turn out to be easy. And what's wrong is we don't know enough. And what's wrong is we haven't been working on this seriously in the past. But I think that's going to change. And I know a lot of the people in the field that are working on this, and they're really good scientists. And we're going to get more. So one of my jobs is recruitment. I started with my own students, because I have more twist, but I think I'm going to try to get even more people involved in it. And with Vicki's help, because it isn't going to work unless NIH decides to help support the work, and I think they are. I'm very appreciative of Francis Collins taking this seriously, and I think he shows real leadership in doing that. Let me just show you a couple of other things. This is a different patient. It was already mentioned by Manny Horning, but it is important. And that is, here's a patient that shows an upregulation from tryptophan, and there's a deficiency in tryptophan. This upregulation is taking tryptophan and going that way, and you're losing your tryptophan. Now, and that's a problem because of where tryptophan else is used elsewhere. And it's used to make melatonin. It's used to make serotonin. And this patient, we decided, must have an infection, and yes, she did. But I just wanted to show you, when you say you have these kind of symptoms, I have an infection and I can't sleep. Well, yeah. It's all set up here for you. It's not strange. This is exactly, and it's not because you're crazy. It's all set up to do exactly that. Biologically, why, I don't know. Oh, this is just, I just want to show you one other thing here. This is why we know that with an infection, the tryptophan is going to go away, and this is just some literature. And I pulled this out, and this has been done in multiple times, but this is data from our big trauma study, and that is when you have, where is it, an infection, you change the expressions levels. I also wanted to show this because it shows you another thing, and that is, I've heard a lot of patients say we really need an animal model. We need a mouse model to study chronic fatigue syndrome. It's a little dangerous. I don't oppose that if someone's really good at doing it, but it isn't something that's obvious and has to be done. And this is the case in the trauma study that we did. Here's the response of human in black and the response in mouse in blue. They're not the same. And in some cases, they go in the opposite direction. So this was a real problem for trauma, because the idea that you had to have a model and you had to validate in a model system before you did the human, if you're going to do a drug that treats trauma, you have to first validate it in the mouse. And that was an FDA requirement. If anybody knows about statistics, the correlation coefficient between the response in mouse and human was 0.06, which means it's random. There were 150 drugs going through the pharmaceutical industry that worked in a mouse. Not one of them worked in human. And it's because it's different. And basically, the take-home message from that, if you have a complex system, mouse may not work. It has a whole different selection going on for it for millions of years. If you have a simple thing, it's likely to work. So we can cure cancer in mice, but that same tool doesn't work very well in human. Cancer is complicated. ME/CFS is complicated. It might work in a mouse, but I wouldn't be surprised that it doesn't. What we're trying to do is to take cells, of human cells from CFS patients, and use those as a model system. And we're first going to try to do it with the white cells, and if that doesn't work, we're going to try to go to muscle, and I suspect muscle will work if white cells don't. Well, I already said that. I want to show you one other thing, because I want to try to deal with the problem of diversity a little bit. Because there's been a lot of talk about must be different kinds of diseases because they behave differently. Here's one pathway, and we know a little bit about this pathway. It starts out with something called GTP. That's a high-energy compound. It's what you use to make DNA. We don't have a direct assay of this, but we have an indirect assay. It looks low in ME/CFS. 2% of the population has a mutation in this enzyme that lowers the amount going through this, and you don't need to know these names. They go down to something down here called BH4, and I know some ME/CFS patients know about BH4, but BH4 makes dopamine and serotonin. And in order to make BH4, you have to have GTP. Our indications are that BH4 is also low in these patients. Now, when you hear dopamine and serotonin, you think, oh, the brain. Yeah, it's in the brain. It's in the gut. It's in the kidney. So we've been looking a little bit at the kidney. And when you have low dopamine in the kidney, you don't secrete salt. If you don't secrete salt, it has all sorts of effects. And so some of these can account for things like low blood volume, restrictions of the blood vessels, can account for a lot of the symptoms that people have. And it's all started by a concentration of that. Why is that low? I have no idea. That's something we need to try to figure. This is very fundamental. This is very fundamental biochemistry of the human body. But so when you see all these different symptoms, they may have a very simple and common origin. Thank you.
Ron, you can tell that that wasn't too bad. Yeah.
Thank you.
Thank you very much. Seriously, we have one or two questions could be asked now, and then I want to turn it over to you guys with a few comments. Yes, yeah, and there's someone right in the back at the top there, Ripia. A man at the back.
Yeah, I wanted to ask, like you said that Whitney was helped by biotin, which he was deficient in. Did you try other things like the inositol was seen to be deficient? Did you try other things and they all help, or did some seem like they should have helped but actually didn't?
The problem with my son is that he continued to go downhill. So we were desperately trying to do things, and as soon as we learned, we can do this, he would get worse. And then it didn't work anymore. It didn't work anymore because just when I found out some of the things we needed to feed him, he could no longer eat. And he had to go onto a PICC line, which is a tube that goes into your arm, threads through, and then goes into the heart. And so you feed through that line. Now that line has to be sterile. And fortunately, I'm a microbiologist, so I don't have a problem with sterility. But it is a problem for a lot of people. And what you put in has to be sterile, absolutely sterile. And there used to be a lot of things you could buy sterile, but not anymore because people didn't want to have the inventory and they didn't want the liability. So you used to be able to buy sterile biotin. Can't buy sterile biotin anymore. So I had a really hard time getting biotin into him. So that's been a problem in terms of inositol. I have a bottle of inositol, but I can't get it in. It's not sterile. I could filter sterilize it, but I think that's way too risky. If we screw up and infect him, he could easily die. I want to point out one other. That's all right. One other little. When we first started with his PICC line, he got a fever, and he went up to 104. Andy Kogelnik, who was his doctor at the time, said if he gets to 104, take him to the emergency room. I don't know what they would do. He was already on a broad-spectrum antibiotics, 2 different ones, 1 gram positive, 1 gram negative. That's exactly what they would do there. He has an IV. We just connect it in. It's easy for us to do that. But we were setting up to call the ambulance, and then I said, I'm going to take his temperature one more time, and it was 103.8. Oh, let's wait a little bit. And it went to 103.6, and then it gradually went down because the IV antibiotics were working. Then he got better. And he was better for about 2 months, and then he got worse again. So I said, oh my God, it was the antibiotics. These things are so broad spectrum, we hadn't ever tried those before. So I got more antibiotic of the broad spectrums and did it again. Put him on it for 2 weeks. No impact. So we've been struggling with what in the world happened. I think we understand. I'm guessing, because looking through the literature, when you run a fever, in order to generate the energy for that, I'm speculating, you metabolize your muscles. Now, this is not human studies, it's some rat studies we saw. And that will give you amino acids. And he can burn those. And that's what made him better. So that's encouraging, though. So that just points out when you sort of look at the data and say with the obvious, antibiotics help next year, must be a microbe. No. It could easily be because he's burning his muscles. Very, very, very indirect. That's why you have to be so careful with interpreting data or over-interpreting data, which is why you have to hang on to all this ambiguity. Don't say anything is known for sure and dismiss things. Don't stop looking for something because that can't possibly be true. Because you could be wrong, and if you're wrong, you're hung up for a long time. Keep an open mind. And what people should be doing research-wise is what they do well. Don't say, oh, don't do that, you should do this. No, if that's what they do well, have them do it, because they'll do a good job. So we have to be very careful not to try to micromanage the research and say, don't do that, do this.
Okay. One at the back, yep, no?
At the beginning of your talk, you mentioned a whole variety of various techniques of measurement, but I assume that most of the slides you showed were based on mass spectrometry. Is that correct?
No. One thing in that list was based on two things in that list were based on mass spectrometry. The metabolites and looking for microtoxins. And we need to do that because microtoxins have unbelievable strong effects on people, and we need to know if they've been exposed. And molds are everywhere, so it's not hard to get exposed. Most of the molds are not harmful, but some are really awful.
I'm very interested because 20 years ago, I attended a meeting of the British Mass Spectrometry Society, and there I found out that the biggest supporter was the racehorse industry. But the problem with mass spectrometry is that in launching the materials you want to measure, you have the danger of fragmenting them, and also the efficiency with which they are detected is very variable. But comparing patients and controls under the same conditions should overcome most of those problems.
No, you have to run controls all the time to do that, which is one reason why I like the idea of going to a professional, not me trying to do it myself. We have a mass spectrometer, but we use it for simple things, or things that we know exactly what we're doing with it. But if it's been a long time since you've done mass spectrometry, it's unbelievable what they've done recently. Sensitivities are incredibly up, and the number of things they can resolve. So we can see the metabolites of the human, but we can also see all the metabolites from the gut. And you can tell a little bit of what kind of microbes are there just by looking at the metabolites.
Okay,
so the instrument, that instrumentation is unbelievable what has happened recently.
Okay. One other question? Sorry? Where? Oh, one in the middle, yes, sorry.
I'm struck when you said that these people aren't secreting salt, because a lot of young people have POTS, and they're all told to increase their salt intake to deal with the POTS. Does that mean then if they have ME and POTS, there is a risk there if they're following the instructions for POTS?
Well, for one, I'm not a physician to deal with that, and that slide was not meant to say everybody's like this, but to point out that you can have these kinds of effects. And in fact, part of the reason for looking at that is that it was pretty clear my son has that. Looking at his salt secretion was extremely low. We can never figure out why he's not secreting salt, but that's probably why. He doesn't suffer from POTS. And maybe that's why he doesn't suffer from POTS, because he's got a lot of salt in his system. So it's very possible that the ones who have that are not having a problem, because they would have higher blood pressure. Don't know.
I can assure you it is a real delight to chair such absorbing, enthusiastic, committed speakers as we've had today. It's really the kind of people I meet in other walks of life are boring, dull, and have nothing interesting to say. You can't say that was the feature of any of the talks today. They were all really worth listening to. Already, I'm supposed to just say something very briefly and then turn it over to you guys about the status of ME research. Well, I feel really messed up because I'm writing a book about science, politics, and ME, and Chapter 3 has got to be completely rewritten now after this conference. I'm sure that J.K. Rowling, Ian McEwan, and other mates of mine like Margaret Atwood have never had to write a chapter after coming to a conference of any type. They may have changed ands and buts and so on, but they've never had to rewrite a chapter again. So thank you very, very much indeed for that. But it shows you how the pace is growing, and that's, you know, interesting stuff's coming along, and there's more to follow. I just wanted to make one quick point, that in my life, whenever there's biomedical research talked about, it's always absorbing and interesting, and something happens because of it. I think that's a feature. And I remember things like Alec Jeffreys talking about DNA fingerprinting, and it was, you know, it was very interesting. Well done. And look at the difference that's made to forensic science in this country. You can take a hair from somebody, you know, who's been dead for 20 years and show that they were guilty or not guilty of something. It's really made forensic science really, really important, because somebody did some abstruse kind of research on DNA fingerprinting. Amazing change to people's lives. The other thing I think about is a student I worked with in Edinburgh when I was a PhD student with him. He was called Bob Edwards, who eventually got a Nobel Prize very late in life for discovering that you could fertilize mouse eggs with a sperm in a test tube, as it was called, but really it was a Petri dish at the time. And it took a long time for him to be accepted by the science community, because he dared to say at coffee once, if I can do it with a mouse and a rabbit, I can do it with human beings. 5 million people have been born now using in vitro fertilization. Despite whatever your political or religious view is, it's a technology that science brought along. And Bob couldn't get an MRC grant, had to go into a private unit in Cambridge to set up the bone clinic there, with Patrick Step to a gynecologist. And nobody would have coffee with him when he was students, because he was talking heresy about doing it with human beings. And things can change from basic PhD work like that. And I can think of other examples. Paul Nurse, who got a Nobel Prize recently, a few years ago, Paul, with Lee Hartwell in Seattle, and Tim Hunt, the bad boy who said naughty things about women at meetings, and there was a journalist in the audience who exposed the poor devil. Poor old Tim. I mean, you just don't make jokes about people at public meetings now, because people are tweeting and twittering and leaking things to the press and so on, and don't we know it? But I don't think Paul Nurse ever saw a cancer cell in his life, but his work was relevant to cancer cells because he worked on yeast and understood how cell cycles were inhibited or increased by isolating the genes. And I think that just shows you never really know where a Friday afternoon experiment is going to turn up, as long as you can repeat it on the Monday, of course. But seriously, you know, you do get surprises, and we should encourage our young people who are doing basic PhD research all the time. I spend a bit of time going around universities, both in the States, talking to graduate students, and in the UK here. And, you know, they've been told by their supervisor, oh, don't talk about politics and the relevance of your work. You know, that's for other people. You know, don't talk about nutrition and the causes of malnutrition. I remember a pal of mine talking about the biochemistry of starvation. And he was asked, what is starvation? How does it come about? And he said, it's not for us in the science department to worry about that. Go along to the arts department or the developmental school, and these politicians talk about starvation and the causes of it all the time. Just understand the fatty acid cycles and so on. And a lot of young people now are looking around for the relevance of their work and need to be encouraged. And when I go to MIT, I can have a row with Chomsky in 5 minutes. We don't quite agree on everything, the anarchist. And but we do excite young people to talk about the relevance of their work, because you don't know where it's going to go to, of course, and what's going to happen, but you should be allowed to think about it. And we only spend a few hours with them, and they get fascinated. Some of them don't want to spend their time at the lab bench. They want to get out into charities and organisations talking about the relevance of science to society and how it affects changes. And they do understand statistics and some of the basic science, and they need to be encouraged. And I think in our field here, we're beginning to see that. We will see more PhD students, postdocs coming along and talking about their work and the things they do. And maybe somebody in an audience like this will suddenly see the relevance of it, which has been missing in academic circles. So we are a team, as it were. We are a group of people who are thinking about these issues, can see the relevance of them and the importance of them, and we've got to really make sure that we open up the channels for that kind of communication. We're beginning to do it with the scientists, and I'd like to see more patients really starting to feel involved. I've been one who's always gone into trouble by saying maybe patients can tell people the kind of science, that they should be doing. And people say, no, patients don't know anything, only scientists understand the second law of thermodynamics and all that kind of stuff. And we have arguments with that like that with usually senior people. But you know what I mean, how important it is that people feel excited, involved, feel they've got something to offer. They may not understand all the details, but they can say, but what does it mean about this and so on? And that's what I've felt in the last few days here. I've never even wanted to go to sleep for about 5 days now, because every minute was absorbing what people talked about. And the relevance wasn't always obvious to Emmy and so on, but it could be. Who knows what's going to happen? Because something's ticking in this field. In your life, you suddenly say something's happening or nothing's going to happen there. But I feel in this field, things have really moved on. And I just want to turn it over to you. But before I say that, I want to say to Pia and Richard, you're simply the best. Thank you very much for the opportunity. For doing that. You made it happen. So the world is moving, and we're all in it to make the world a better place for our families and grandchildren and great-grandchildren, whatever age you are, Ron. But you know what I mean. And the commitment is just amazing. So I don't see it in many other aspects of life, but I certainly see it here, and thank you very much for the opportunity. I shall never stop either, and I'm sure other people will. So thank you very much.